When people think of cloning, what often comes to mind is some scary, hidden lab filled with humans floating around in capsules. But cloning has actually been around for millions of years, and it's not nearly as scary as what the movies have made it out to be. So, what even is cloning?
Cloning is defined as the process of generating a genetically identical copy of a cell or organism.
Believe it or not, cloning actually is a naturally occurring process. During the S phase of interphase prior to mitosis, a parent cell's chromosomes are duplicated before later dividing into two identical daughter cells. On a more complex scale, we have twins.
A fertilized egg splits into two embryos, thus resulting in genetically identical fetuses. There are three types of artificial cloning. Gene cloning produces copies of DNA, reproductive cloning produces copies of animals, and therapeutic cloning produces embryonic stem cells. In gene cloning, scientists locate their gene of interest, the part of their DNA they're interested in, and find a suitable vector for it, usually a plasmid. Using restriction endonuclease, they cut both the GI and the vector at the same location and then paste the GI onto the cut vector via DNA ligase. This forms what we call the recombinant DNA.
They then introduce the recombinant DNA into suitable host cells. Those that successfully take it up are referred to as transformed cells. As they multiply, they make more copies of the DNA that scientists are interested in researching.
In reproductive cloning, researchers remove a mature somatic cell from an animal they wish to copy. They then transfer the DNA of that cell into an enucleated egg cell. The egg is then allowed to develop into an embryo in a test tube and is later implanted into the womb of a female if successful. Ultimately, the female gives birth to a clone.
The process is known as somatic cell nuclear transfer. It was first done in 1996 after 276 failed attempts, resulting in Dolly the sheep. The reason it's so difficult to pull off is because combining two different cells perfectly without making any mistakes requires an extreme amount of precision. In fact, only about 5 to 15% of the transferred embryos result in live offspring. Therapeutic cloning has a similar process to reproductive cloning, except without the implantation. Scientists clone the embryonic stem cells in the labs, and they use that for all sorts of medical innovations and research purposes.
Cloning has served the life sciences so much more than merely copying animals. In 1952, Robert Briggs and Thomas King discovered the concept of nuclear differentiation during the very first cloning experiment documented. It has enabled us to study different phenomena in biology to an unparalleled extent. It's an innovation that will help us discover more in the future.